let's continue from the previous class. So we are going to discuss about the components we have to consider in our visualization. So one of the main factors you have to choose for your data visualization is which plot types you're going to use to represent your data set. So one of the easiest things you can choose is the nominal comparison. So this means that it is a simple comparison of the quantitative values of subcategories. So let's say if you have different groups like the cycle group 4, 6, and 8, then you can basically plot how many items or entries belong to each group. So here, cycle 4 has the 12 counts, and the cycle 6 has the looks like 7, and 8 cycles has like 15. So that information you can basically give the two layers of information. So first one is the number of items, and then the how many groups you can define to comparison. So the bar plots in these examples we use many times for your data exploration, and that will be probably the first trend you can think about the visualization. Okay, so another types of visualization we normally use is the time series. So literally, this is basically comparing the information flow by time changes or procedures. So here example is the popularity of American names in the previous 30 years. So from 80s to 2000s. So looking at those year range, you can check the numbers or quantity. So time series is if you measure some gene expression over the experimental times or developmental stage, you can also use these types of information, right? So this is quite intuitive way to represent your data set. Another commonly used the data visualization form is the correlation. So if you have more than two data points, correlation is quite useful. So what I mean is if you have two linear relationships such as like x-axis here and y-axis here, then you can represent that information using y equal x, and you put the constant and the intercept. Then you can say the xy relationship forming certain information. But in real world, if you measure some data set, you always have more than one relationship. So if you, as you look at here, you have relationship between A and B here, right? But you also have the relationship between the B and C, right? So all that information you might want to represent the correlation in the one space, right? So that case, you can only put the coefficient value from the correlation calculation, then you put it into the scale, then you calculate the numbers, right? So think about in this situation. So if when you measure gene expression from certain tissues or cells, the think about the number of genes in our genome. So human has 60,000 genes, right? We have 20,000 genes for protein coding and 40,000 genes for non-coding genes. And those genes have the relationship with other 60,000 genes, right? So if you want to plot that information using this linear relationship, you might want to have like a 60,000 times 60,000, right? So you need to huge number of spaces to represent that information. But here you have such a neat space, only putting the correlation values into here. Then you can say, the first and the second items correlation or the second and third items correlation, you can easily say which pairs turn out to be highly correlated between pairs, right? So you don't have to put all the 60,000 square numbers, right? So like we see in the gene expression information, you might find correlation in the patient phenotypes or any large number of items and measurements. So that case, showing the information in correlation is quite useful, right? So and then the correlation also used for some situation like when you want to find some patterns within your data set. So for instance, like if you look at the 60,000 genes, and some of genes should have higher gene expression by themselves. So what I mean is, if you have the 60,000 genes and some of the genes has higher correlation to each other. So for instance, like 200 genes out of 60,000 genes have higher correlation to each other because they're working in the same pathway. So that case, finding sort of those patterns. So what I mean is, finding some genes or some sets of genes having higher correlation that represents some meaning in biological context. So here I have like 200 genes, they have the high expression, and those genes should have working in the same pathways because they are interacting together and they appeared in the same context. So they should have higher gene expression than the correlation than others who are not in the same pathway. So what I mean is, let's say, if you look at the cell cycle pathways, if you look at the cell cycle pathway, so this is one example. So this is the how cell undergoes cell preparation and cell cycle during the mitosis, and this is the sort of the diagram. We normally see cell cycle like this, but when you plot those protein relationship, then you can summarize the relationship like this, right? So this is sort of the consequence, and sort of those area they initiating the cell cycles, right? I think this is more proper one. Let's click the pathway image. Okay, so this is the cell cycle pathway. So there are some initiating points like a TGF-beta or whatever P300 or the what is the SMC? SMC3. Yeah, the protein working cohesin, right? So the mitosis has to make a junction, right? So all of that information initiating the signals for the cell cycle process and mitosis, and those cycles, they have sort of a flow from the early points to its downstream consequences, right? And it has the kinase information, and it has the targets that operating the cell cycle conversion and phase transition, and those information has highly dependent to each other. So highly dependent to each other, in other words, in the mathematical terms, we can say that this is highly correlated to each other. So what I mean is, if you have higher gene expression values of CDC25A, then you should have higher gene expression of CDK2, because those are connected to each other, they interact to each other, they influence into each other. So if you measure some higher gene expression in this gene, then that means that these genes also have high expression as well, because they're working together, right? But if you look at these gene numbers, this is only like on 100 genes something. But as I mentioned, that we have like on 60,000 genes, right? So what are the rest 59,000 genes locating, right? So those genes now located in cell cycle, they are working in a different pathways. So if you measure the correlation value between this and this, then you will see much higher correlation value between these compared to these and other non-cell cycle genes, right? So that means that if you're measuring the higher correlation in a biological context, then one thing you can infer from that information is that those protein might be working in the same pathway or in the same situation, like, in the same developmental point or same organs, right? So that case, we utilize the information about correlation, right? So this is also one way you can represent the data properties. Or one more thing we can think about is the ranking. So when we get some information, especially the quantitative information, we can make a rank for each item, right? So let's say if you have gene expression of entire genes in human genome. So let's say you take the lung and those lungs from the cancer patient, and if you measure the gene expression out of lung cancer patient's lung, right? Then you get some numbers that, in this case, the numbers has to be related to the gene expression, right? So that number should be the number of mRNA copies, right? Because the gene expression means that how many transcript expressed in that tissue or in the cell, right? So if you measure the gene expression, then you basically measure that how many mRNA transcript you can count from that tissue, right? So not all the genes have the same transcript numbers because, out of 60,000 genes, there must be some genes have higher expression in lung, right? So which genes should have high expression in lung? Probably the genes working in lung development or lung maintenance, those genes should have high expression in lung, right? So likewise, if you look at the thymus is the place for the immune system. So the genes involved in immune system should have higher gene expression than others, right? Or let's think about the brain. If you look at the prefrontal cortex, which is one of the major organ of the brain function, then if you measure the gene expression, then you should have higher gene expression working in neuronal cells, because the neuronal cells are the major cell types working in brain function, right? So basically, if you measure gene expression from your experiment, then if you want to look at which gene expressed higher in my experimental samples, then in this case, you can basically use the concept of ranking, right? So ranking is comparing values in relative magnitude, right? So this is one example of how you represent ranking. But I don't really like, the circular format for the ranking system. Basically, you can plot in a bar plot rather than these flamboyant types, circular plot. But anyway, this is just an example. Okay, so one thing we can say is go away. Okay, let's say okay, so we looked at the SMC1A, then we searched that. Let's look at the data together. Okay, so we will look at the SMCA protein expression in a human protein atlas database. So you want to know about which organ or which tissues have high expression for this gene, right? So there are some information about tissue expression. So you can click that tissue. Then you can see expression of RNA. This is the gene expression and expression of protein expression. So in a RNA level, which tissues have high expression? High expression comes from bone marrow lymphoid tissues. So we can say that these genes probably work in some immune cells as well. How about the protein? Protein has the high expression, but we cannot distinguish the rank. It's probably the ranking it's only like 3 tiers, higher, medium, and low, because they have the same scale like this, right? So we cannot choose the highest value, the top rank for this gene, but at least we can say the top ranks genes, right? Okay, so how about expression according to functions? So what is this gene? This is the adaptive immune response. So the genes is probably working in a T cell information. Okay, so here is the rank. So gene expression overview. So this is just a quick overview, and this is more detailed data set. So if you look at the gene expression, the highest value from the thymus, right? So we can just use the bar plot and showing the rank between tissues, right? So we can rank from this one and this one and this one and this one order. But for the visualization point, you can sort it by the expression by tissues is much better for expression, right? So here, yellow colors are brain, and light yellow from the visual system. It's close to brain, but not really brain because it's retina. And what about the thyroid gland, the peripheral gland system? And what about the intestine and the stomach, the digestion systems and reproductive organs and the muscle systems, right? So we can see the rank and also by groups, right? Okay, and also if you look at the protein expression, okay, so protein expression also high in thymus. So you can search by this. Okay, so this is how you check the rank information for your information. Okay, so we have another visualization strategy to show group. So groups normally has discrete information or categorization. And one of the things you can use is this the tile style categorization. So if you look at the graph on the left, this side, this one contains 4 groups in this plot, right? So you can clearly see that there are 4 groups. So first one, second, and third, and fourth. Then you can see that there are some subgroups, right? So subgroups like it's actually 3 groups, right? 3 groups, 1, 2, 3. So blue, green, and red. But I don't really like the blue colors. But anyway, and then the first group has the 4 subgroups, and the group 3 has the 3 subgroups, and group 2 has 2 subgroups, something like this. And you can say there are some structures about the groups and subgroups, but also you can see the quantity, right? So you can compare the quantity between groups, right? So this simple rectangular type information, they have they have multiple information about the groups and also the quantity belong to each group, right? So one of the most useful things for this categorization and quantity information is genome tile. Sorry, this is not mosaic plot, right? Okay, so these types of plots we normally call mosaic plots. So this looks like this, right? So if you have a single bar chart, it looks like this, and it has the one dimension of group information, right? So one dimension means that it has these 5 subgroups, right? But sometimes you have two dimensions of groups, right? So you have the same 5 subgroups, but and then you add up the 3 different groups, right? So it's basically 5 times 3. So that case you can use the stack bar chart, but if you use the mosaic plots, then you can represent the information like this, right? And you can add one more information, use the mosaic plot as well, and that is also quite neat. You put it in one chart, right? So if you are interested in mosaic plot for your portfolio or for your practice, it's very good to have for your exercise. Okay, and another way we can represent the groups is the hierarchical plot, right? So if you look at this graph has the group information 1, 2, 3, 4, 5, 6, 7, and then you have the subgroups, right? So in each group, it has like a 7, it's all 7 subgroups, right? So you can also represent that information like this, right? So let's say, draw, it has the 1, 2, 3. So you can basically draw the 3 branch. And how many subgroups? 1, 2, 3, 4. Then you can say 1, 2, 3, 4, right? And what about the group 2? It has 1, 2. So you can say like this. And what about the group 3? It has 3, 1, 2, 3, right? So these mosaic plots can be used as a hierarchical plot, right? Hierarchical plots are including that information for the major groups, but also the subgroups as well. So why we have to think about the hierarchical groups? Hierarchical groups is used in many cases. So let's look at one of the examples. Hierarchical plots, in other words, we mentioned that the dendrogram. I like this one. I like this plot. Oh, damn. We have to pay it. So let's look at the free one then. No free things. Broken academic system. Single cell dendrogram. Okay, that will be great. Okay, so if you look at this information, so this is the information about when you measure which tissue, that's probably one of the immune tissue. Then you look at the cell information based on their expression. Then you make a relationship between the cell types, right? So how many cell types here? B cell and K cell and gamma delta T cell. and CD8, CD4, neutrophil, and monocytes, and macrophage, and active macrophage, right? And then you can also see the relationship between the cell types. So macrophage and active macrophage, they're all from the macrophage. So they are in the same branch, right? So this clade or branch. Then the macrophage is basically derived from monocytes, right? So they are in the same branch as well, right? So monocyte macrophage and activated macrophage, as you learned in the general biology. And the neutrophil also from the myeloblast, so they are in the same branch as well. Okay, so what about the T cells? T cells from gamma to alpha here, and it divided further by CD8 and CD4, and that in the same branch as well. And the B cells are from the same lymphocytes, but they're taking the different branches as well. So all of that information is represented by the hierarchical dendrogram. And in that case, we can clearly see the information about how they make a branch and how they make a relationship to each other. But this hierarchical plus doesn't have the quantity information. So when you represent the lineage or when you represent the cell type relationship, that would be very useful. But quantity is lacking this type of graph. Another way we can think about the dendrogram is this. Let's look at the information like this. Okay, that would be great. Okay, so this shiny graph means that the clustering of gene expression, right? So it measures some gene expression from some samples. Here is the cells, right? So this guy measured the gene expression of some 100 genes. And how many samples? Some tens of samples. And this is the cell 1 with some treatment or some condition. We don't know that. But anyway, they put the cell 1 to 3, group 1, group 2, group 3. That means that they have different conditions or some characteristics, right? And that means that the cells should have some gene expression uniquely expressed in this gene, in this group, right? So this group has high expression 2, and this group has the low expression, so -2, right? And they call this is class 1. And what about the cluster 2? These genes, so each lines on here, let's zoom in, its lines here. These are the individual gene, right? Individual gene on here. Okay, let's zoom further. Right? Okay, this is the individual gene, right? So this cluster, the sort of these genes, how many? 1, 2, 3, 4, 5, blah, 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 10 something. Those genes are lower expression, -2, the cell 1, right? In contrast, cell 3, the group 3, has the higher expression, right? So these clusters has opposite patterns. So what about the cluster 3? Cluster 3 has a high expression on group 2, right? So this is uniquely highly expressed in this group, right? So in this case, we also use the hierarchical dendrogram like this, right? Like this and this. So we have 2 dimensions, 1 dimension and 2nd dimension. So what is differences? So first dimension is representing the relationship between cells based on gene expression, right? So how similar, the gene expression between samples, and they make a cluster, they make a membership. But if you look at the second dimension, this is also based on gene expression, but then they make a cluster information between genes rather than samples, right? And it has some relationship like this and this, but in a broader sense, it has the membership like this, right? So sometimes there is a cutoff you can define the clusters between items, but sometimes it's not. So you have to look at by your eye, then you have to make a decision which boundary you have to go for these clusters, right? So this case you can use the plot type called heatmap, right? So heatmap means that it has the quantity information and it has the two-dimensional information as you see in here, right? It shouldn't always have to be like, samples and genes or something. I mean, any kinds of, two-dimensional relationship, you can put it on a heatmap, right? But in our situation, biologists, we normally use the samples and gene expression, or samples and some kinds of the expression, right? So one of the most useful way to do this heatmap, the package called complex heatmap. This is quite useful, and it has a free gift book, right? So they have free textbook. So if you search complex heatmap, then you can go to the website written by Joe Kogu. I think he's one of the crazy guy in our lifetime. So let's go to his profile. Oh, where can you find him? I mean, we have to look at his face because he's just a crazy person. So, I mean, it's worse find the guy. Oh, Jokoku, MIT. I think he's in MIT. Then visualization. Circular visualization. I think it will lead to his website. Come on, Dr. Gu. Okay, so his name is Ju Guanggu. He is in German. He's in one of German institute. I'm totally impressed by him. He's amazing guy. Cannot find his PhD. Yeah, this guy. I think this guy. No, he's a student. Where can I find him? I think I saw his face somewhere. Yeah, his tool is very impressive. Because it's always maintained and also bring quite useful information about the our visualization and genomic data. And sometimes when you learn something, it's always good to find good person. So you have to track down his record and his whatever his outputs, his products. And if you follow their the packages or papers, you might find the quickest way to learn some topics. And not everyone can do that. So what I mean is not everyone provide such a good information, but sometimes there are some distinct people you might have to remember in your learning. And one of the person is definitely I can choose this guy. This guy is very, very impressive. Anyway, so if you are keen on to learn complex heat map, then go to there. Is his GitHub page. Okay, this guy. Yep, this guy. Yeah, I mean, this guy is just a crazy. Yeah, I follow him also. I follow some of his packages. I mean, definitely this guy is very impressive. And another impressive guy, just TMI, because we are on the YouTube, so we don't have the time limits. Sigmina GitHub. And this is another guy making impressive package in genomics era. His name is Xishang Wang. I think he's based in China. And he also have some very nice book about genomic analysis. And this is the classical the topic in cancer genomics, which is the mutation signature. This is how mutation happen in cancer patients, and there are any reason to have that cancer mutation, right? So if you search cosmic mutation signature, then it will lead to the web page based on UK, the Senya Institute. I mean, you might heard the institute name. This is the best institute in our field for the genomic research. And if you click the information about mutation signature, then you will have 4 different types of signatures. One is SBS, DBS, ID, and CN. SBS means the single base pair substitution, which is equivalent to SNV, right? And DBS is double base pair substitution, and ID is insertion and deletion. So these two are equivalent to index we learned in genetics. And DBS is, I mean, as I mentioned, that this is double base pair, so it's only 2 nucleotides, so it's shorter types of index, and this is just rest index. And CN is the copy number changes. As we learned, this is like a duplication or deletion, right? So if you click SBS signatures, you have like 100 signatures explaining each cancer patient, right? So if you look at the SBS1, and this is the mutations happen mostly in C to T change. So mutation happen in C nucleotides and it changed to T, right? So what is the possible biology? So in other words, why those patterns mutation happen in cancer patients? That means that the endogenous mutational process initiated by spontaneous or enzymatic deamination, blah, 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 right? So it's spontaneous mutation occurrence, right? So which means that, mutation occurs randomly in our cells over the lifetime. So that case, the mutation has some signatures in our DNA like this, right? So what about other types? So what we know about the cancers? So let's say lung cancer, we know that the smoking is a problem, right? So what about the smoking? So if you do smoking, then the mutation happen in this way. So this is possible etiology, means the possible cause. This is the tobacco smoking, right? So if you see the cancer patients who have severe smoking history, those patients have the mutation like these patterns, right? Or there are some other issues like a recombination repair failed, or a POBEC family deamination happen, or sometimes DNA mismatch repair or UV exposure or something. So why we have to care about these mutation signatures? Because cancers have so many genes causing disease, and those cancer genes are overlap between different cancer types. So you have to dissect mutation types and you have to think about the cause of the mutations, right? Then you can develop the treatment, or you can develop some intervention way to prevent those cancer prognosis, right? So one way we can do is using these patterns. And we have the database information, so you can use this information for your research. But this is quite old topic, so this is more than 10 years. So in the meantime, people develop some nice tool to use the signature database. So this is the package developed by, what's his name, Shishang Wang, and he proposed how you use the data set and how you draw the plots from the data set as well, right? So this is how you process that, right? And all codes, and this is again all written in the public format book, right? So not really, you don't have to pay anything. So you can go to this link, and if you are interested in genomics, then just install and just follow the code in your RStudio, right? So I mean, why not? So there's a heaps of things you can learn from the internet, and definitely Sigma is one of the things you can find useful. Okay, so that was a bit of TMI when we explain about the hierarchical relationship. Okay, so let's go further. So we can also represent the relationship using the proportion, because many of the information we are dealing with is the quantitation. So if you have the information about the quantitation and categorization together, then you can use different types of plots, like, as we discussed, bar plots, where bar plots has the different orientation, or bar plot has the group bar plot, stacked bar plot, or stack density, or mosaic plots, or you can use the parallel sets, like, if you have the time series, and then you can use the flow of the quantity between groups, right? So you can use all of that information. But we can also use the pie chart. But pie chart, I would say this is one of the worst way you can represent your quantity information, because pie chart doesn't have very needy the layout to represent the quantity information, because you can only have one dimension of the quantity. So let's think about, if you draw some pie chart information, it's basically replaced by one bars in a stack bar chart, right? So there is no point we have to use pie chart. So you might want to use multiple pie chart, but this is again gross, because you can, if you use the stack bar chart, you can basically put these three things into the stack bar chart. So there is no point you use the pie chart. So there is some joke or muck arounding that, if you use pie chart, you don't know anything about visualization. So don't go that route, because this makes you mature to visualization field. So pie chart in your brain from now on, forget it. Okay, so and also if you have some information, we're talking about the distribution, and this is the useful, especially when you think about the data property and the models of your data set. So the situation, like, if you choose the statistical evaluation and you have to come up with some information about your data and data distribution. So in that case, we have to use some visualization format to plot your data distribution. And one of the easiest things you can try is the histogram, right? This is quite familiar format from your previous education. And density plot is the also useful information. This is the information you are based on the cumulative functions. So you have the quantity information from the 0 to 100 scale, right? 0 to 1 scale. Then you put it into the cumulative functions. Then you can calculate that density. And in other words, the density plot is also transit to cumulative density because this is basically the same idea. So from 0 to 1, then each intervals you have some probability functions. Then if you add up that information, in other words, if you aggregate that information, it has to ends up 1 in the end, right? So within the entire range, you have to have the probability summing up to 1, right? Or you can use quantile plot. So if you have the expected distribution and the each observed variables from the from your experiments, and you put it into point, right? But in terms of distribution in your stage, the easiest one is the histogram and then density plot. So if you fully utilize these two plots, I think that is way sufficient for your distribution exploration. Or other types of distribution plots we can think about is the box plots, right? So box plots is also one of the famous way we can put the distribution comparison, right? But compared to histogram, the box plot has some advantages, right? So one is that it has the quantile information, so you have the median on a in the middle, and you have the first and third quantile, which is 25% and 75%, and you also have 95 quantiles for each end as well. And unlike histogram box plots, you can put multiple of them. So here you have the 3 different groups for the box plots, right? And you can put it into one plot for the 3 information. Or you can use the violin plots, or you can use the strip chart, you can use the CNAP plots. All of that information you can also use for distributions, right? I think these plots, you can use for distribution, not just the exercise, but also for your portfolio, because this is very useful to make it. So if you go to this textbook, there must be a way you can visualize the plots. So there's no code. So that case, how can you search STDHA? The website I recommend you, right? STDHA violin ggplots. Then if you search that, so let's say if you want to draw the violin plot using ggplot2, then you can search that information and go to this site. Then there are some easy comment. You can draw the violin plot in a second, right? We'll cover that one in soon. Okay, and we talked about the distribution, and now we can talk about the deviation, because the deviation is how far your data points of your interest located in a distribution. So deviation is the information you can infer from your distribution. So once you get the distribution, then you can say that the data points of your interest and how that appeared in the expectation. So what I mean is, let's say if you measure the gene expression and that gene expression is normalized in this way, so you can say that the mean value of this expression is around 0, right? According to normalization. And then gene expression is low in here, low, and expression is high, right? And then you put 100 samples on here, right? So each bar containing the frequency of samples. Oh, sorry. This is more than 100. It's probably like 10,000 samples because the frequency axis is quite high. And then your samples of your interest, let's say you want to, you basically calculating the gene expression of someone who took some medication, right? And you want to compare the expression of certain gene in a general population. Then you can get that the samples you're looking for is located in here. Then you can say that deviation from the population mean, then you can say how unlikely these samples has the expression by the medication, right? So deviation is one of the points you can think about the statistical evaluation, statistical chance, right? So deviation used in that concept. And also deviation also used for group comparison. So if you look at the groups, like you do the high treatment and low treatment, then you can basically compare these two groups and how the groups has different median. So median on here, right? So these thick lines mostly represent the median. So it has or upregulated some values in a low treatment group, right? So that case, you can also think about the differences. Or you can also think about the deviation between groups. So you have the 5 different groups and it has some distributed patterns. And you can see that this group has a bit of different the values compared to different 4 groups. There must be reason, right? So you can guess that the reasoning out of these distributed patterns, right? Okay, so what would be the good things you can think about this deviation? So we have portfolio information, right? So you have the portfolio papers and go to supplementary table, and there are some information about gene expression, information about copy numbers or information about the protein expression. Then you can make a plot for the one of the gene expression, and you can compare that expression with other gene. Or if you draw the distribution of certain samples, then you compare that information with other samples. Then you can see that there are some differences between samples, right? So in that case, you can also use this deviation concept. Okay, so we have many different ways to look at the data. So for each purpose, you have to think about the right figures or right plot chart, right? If you want to go to comparison or look at relationship or look at the composition or look at the distribution, and each of them has some, the decision points you can make a good visualization format. Also, one thing you might want to consider in your visualization is color and also alpha, right? So why color is important? Because color itself, it represents the group information. So it has the 3 species on here. So they are on all same points, but you can distinguish them by colors, right? So let's think about this way. So here you have three species, Setosa, Vescolar, and Virginica, and this has different colors like this, right? And you can also use different approach to distinguish species. So let's say you're not going to use the colors. You can make all black colors for the point, the data points, but you have different shape for data points. So let's say you want to use Setosa as the triangle, and Vest color, you can use the star, and Virginica, you can use the circle, then you can put it on here, right? So you replace circles to triangle, and this color is the star, like this. There is another way you can make a distinction, but I don't really like the making different shapes for the group definition, because the shapes is less intuitive than colors, right? So how can we know that? So this one is drawn by the colors, right? So let's try it together. So let's make and see together. So let's go to here. So iris, this is iris. Library, tidyverse. Come on, tidyverse. Oh no. ggplot, iris. Okay, so these days we're going to use this, right? Yes. I don't know what's in there, so to use that. Okay, so what was it? The sepal length. Sepal length from here, and sepal width from here. Okay, then geom point. Yes, colors. And pieces. Okay, let's look at this together. Oops, no, colors is color. Okay, so this is one way where we can try shape like this, right? So I want to show this one and this one together. Two plots P1 plot grid P1 plus P2. Oops. Plot grid P1, P2. Okay, let's look at together. Okay, so this is basically same information. On the left, we use the colors. On the right, we use the shape with our colors, right? So what would be the good thing? So how can you make a conclusion in a second by this figure, right? So if you're both, and if you want to sell this image, this conclusion to your customer, which one you're gonna use, right? You're not gonna choose this one because this is not, one-shot conclusion. But this is like you can get a sense that from these colors in a second, right? So you're not gonna use this one. So as I mentioned, the colors, the humans are visual creature. So using different colors is always better than using different shapes, right? So this is another example. Okay, so colors used in that way. And we can use one more thing, which is alpha. This is another word we call transparency, and it has the scale from 0 to 100, right? So why we have to use alpha? Because it can showing you continuous information. It can show the range. So for instance, like a correlation. So correlation efficiency has the range from -1 to 1. So if you use the alpha, then you can see some changes between coefficient for the correlation values. So in that case, it will be extremely useful. And if you want to use colors, there are always good choice for colors, right? So ggplot give you basically the ggplot default colors. So if you use ggplot, it always choose 3 colors in this order. But there are some pretty way you can use the color palette. And one of the easy things you can go for in these color palettes, but sometimes I want to find better colors. So that case, these days I found very pretty things because I really like colors. So I used map brewer package. It is beautiful, right? Because they have color schemes like Archambault, Austria colors, right? Many different color schemes. So if I want to choose my colors and I choose one of these things, right? So I can go for that, right? So there's so many nice color scales out of these packages. So this is also up to you. So everyone wants to use the beautiful colors for your visual output, right? So you don't want to make ugly things, right? So choosing the right colors and choosing the good color is the critical things for your visualization. Okay, so we can see some examples about using colors. So this is one example. We plot the location of the gene on the chromosome. So this is probably chromosome 1, and you plot that the location of the gene, and I use the color codes for the different gene type. So some genes are protein coding gene, and some gene is microRNA gene. You know about microRNA, right? And some genes is rRNA, some gene is the immunoglobulin genes from the immune system, and some genes from the small nuclear RNA or other gene types, right? And what is the bad things about this plot? What is the problem of this plot? So the problem is there are too many colors, right? There are too many colors from red to purplish. But if you look at this plot, you can't really see that which gene type located in where, right? And if you look at some of the gene type like miscellaneous RNA or pseudogenes or protein coding genes, these genes are, in terms of functions, they are very different genes, but you cannot distinguish the colors between them, right? And also the gene, like immunoglobulin genes, they are in the same functional category because they are from the immune system. So you can, at least you have to put the groups like a red, or reddish something, right? Dark red, light red, or something red, violin red, something, but it's not, right? And these genes are small RNA, scRNA, SNO RNA, snRNA, microRNA, long non-coding RNA. These are small RNA or non-coding RNA, so they have to cluster to each other. And pseudogenes, they have to cluster to each other. And protein coding genes are totally different from others. And you have to make some, color scheme to represent the groups, and also has to be simplified, representation of different gene types. But this plot is failed to do that, right? So why I bring up this example, if you are doing portfolio or if you're doing visualization, some of you might follow these mistakes as well. So you have to think about how I can choose right colors, how I can choose the simplified scheme for the color usage, right? Otherwise, your colors is like a too diverse and no one can understand your colors, right? So the point is that using colors or using better visualization, why we do that, we want to be read my portfolio or my outcomes by others properly, right? So you have to keep you have to keep thinking that how my outcomes, how my figures, how my plots will be seen by others, right? So that is the critical things. So one of the examples about the color choice, this is the color information about the model region, right? And this is the west, south, midwest, and northeast, and the color code used by different bars. And that one looks fine. That one looks fine. Oh, why I put? Did I put it for the wrong examples or not? To me, it's fine. Yeah, I mean, it's okay. Okay, so let's look at more examples. So in these days, when you represent the continuous information, you can use the alpha, but also you can use the Verdi scheme, right? Verdi scheme is it choose the colors from the lower scale to higher scale using that color code from the yellow to purple or yellow to through the red and purple, right? Yellow to red to blue, yellow to red to blue, right? So these types of color scheme we call varieties, right? Varieties is quite useful. So this is quite nice. This is quite needy. And if you want to, let's say, this is the how many Austrian has schizophrenia diagnosis by month and year, and we can easily think that the people between 50 and 60, and those people around the these time points, they have higher rates of schizophrenia diagnosis. And can you see seasonal differences? Probably yes, around the July, around the August, July. In Australia, this time is the winter, right? So it's not summer because it's South Hemisphere. So they have higher schizophrenia onset around this time, right? So that is one way we can think about the continuous information. But this continuous information, why we do care about the Verdi scheme? So, or the magma scheme, plasma or inferno, these schemes are good. But sometimes some journals, they totally bad at the color scheme. So let's look at this. This is the color scheme for the hot weather in summer in Europe, right? So this is France, this is UK, German, Poland, and Spain. And can we guess that which country has the highest temperature in the summer, right? So let's say my expectation is the dark red is the highest temperature in this figure, but it's not. So this is the lowest one, and orange, red, dark red, magenta, it's fine, light magenta, purple, light purple. This guy is crazy because the color scheme has to be light to dark, right? But it's totally fair to bring that message, right? So we don't know which area is the hottest temperature in this plot, right? This plot, this visualization came up in one of our major journal in Korea. Major journal, major newspaper, they totally failed. They don't know how to put the numbers. They don't know how to use colors. They are way soccer than the elementary school students, right? So this is very bad. So there is no, bottom line to use the color schemes, right? So, but if you look at the newspaper from now on, from this perspective, you will find there's so many bad visualization. There is no basics about the visualization. There are so many journalists totally fail at the visualization in our country, right? So how we can bring right information to the public, right? So one message is that if you're doing really great in the visualization, then you can do way better than those journalists. At least we can, I mean, if I were customers, I would open my wallet to those journals, right? But in our countries, we have so many bad things about the visualization. Oh, I mean, this page is totally gone. I really like this page, but yes, there are some collection of bad visualization from the news media in Korea. Yeah, but there are so many things, so we don't have to point out that. Okay, so one more thing we have to care about is the colorblind, right? So there are many people in our society, they cannot see the colors as we see, right? This is not the bad thing. This is not the correct one. We are just different because, we born different way. So there are some strategy we can use the color code for this colorblind or color friendly way, right? So why we have to care about this? I mean, this is not just political correctness. This is for your readability. As I mentioned that why we make what is the aim of visualization is we want to see our outcome, our plus to people as many as possible, because I want to expand the audience to bring my visualization. I want to sell my idea to others as many as possible. So in that context, we have to think about the colorblind schemes, and we have to make the plots with those schemes, right? So that case, you can choose these color schemes, and there are some codes you can use for your visualization. Then if you practice, choose those codes. So you click this link, then you can copy this color code and use for the your visualization format. So this is another way you can consider. So one more thing you have to consider is the font, right? So choosing right font is important for your visualization. So this is the example from Apple website. So on the left one is the font from the Apple, right? iPhone 13 Pro. And this is the way, if you use just the Dodum Che, whatever that fucking font, sorry, the very boring font for our the documentation, right? It's a free one, but no one really wants to use it. And if you use that, it's less attractive, right? So no one wants to use that, right? So this is again, this is from Samsung, and they use the right font. And on the right is just one of the Dodum Che again. Then the the visualization is very gross, right? So having great font is important, right? So these days, our computer has default font with the amazing choice. So if you use the Mac, if you choose the Mac, then the default font is Helvetica, right? So Helvetica. So this is Helvetica, right? Such a great font, right? So the Steve Jobs, he spent a lot of time to develop this font for Apple product development. He thought that the font is one of the major components for the computer and the mobile phone development. So he spent lots of time and money to find this font, right? So Helvetica, how it came. And Windows has really great font as well. I'm not sure whether I have, because this is Mac. These days, Windows has, it's called, I don't think it's installed in here. It's probably installed in your computer. It's called Media Pro. I really like this font as well. Yep, this is the Myriad Font Pro. This is also very neat, right? This is very, very nice font, right? This is very right space between characters, right? So this is good as well. So having great font is useful making your visualization outcome, right? But in reality, you don't really have to care about that, because your RStudio and your ggplot will use system font, which is the default in your computer. So if you use Mac, that will be automatically Helvetica. If you use Windows, that will be automatically Myriad or Arial. So you don't have to really worry about that. So you can use just the system font, right? But if you do sort of other visualization work in your future career, then font is one of the things you have to consider. Okay, so this is one example, and this is this is the website. You can choose for the colorblind colors. So let's say if you want to use the diverging color scheme for the 3 class, then you set up here. Let's say we want to use 5. Then you can choose the color schemes. Then you see whether that is colorblind friendly or not, right? So you can click this. So this is not colorblind free. It's not friendly too, right? Or you can use 7 as well. It's working. Or you can use the qualitative, and this is the discrete information. And I don't know, this is probably better way. Colorblind safe. I don't think it's really for the qualitative more than 4. Right, it's 4. Okay. So that is the one way you can consider. Okay, so this is another consideration, which is the scales and axes. So scales, as we know that this is the unit for your data points, and axis is the x-axis and y-axis. So how you put your information in which dimension, right? So this is one examples I get from the calling bullshit. So you can see that literally something bad going on here, right? So this is the data about average number of actual weekly hours of work in main job full-time according to 2013. So if you look at here, if you look at just 1 second, then you can get the Romania they have company long hours for working compared to France, and some people think that Romania is worst and France is great. But if you look at the scale is quite tiny, 36 to 42. So if you work 41 hours per week, 37 hours per week, it's just a 1 hour differences in terms of the week scale, right? So this plot is intendedly showing the scale, and it brings some intention, and it is toward the information to make some conclusions, right? So if you put the 0 to 42 scale, 0 hour to 42 hours, then the conclusion will not be same as you seen here. Okay, so this is the way. Okay, right? So Romania and France, they work pretty much same hours per week. And this is another bad example. So this is the comparison between oxytocin treatment groups versus not, and how they make changes. And they distorted the y-axis case from 0 and quantum jump on here, and they omitted between 0 and 0.8, and they only showing 1.05 to 0.8, and they showing that drastic changes between these groups, but it's not, right? So that is the another problems if you intendedly distorting the information by wrong scales and axes. This is another example. So this is the examples about the length of genes, or how long your gene per chromosome, right? So x-axis is the chromosome 1 to chromosome Y, and y-axis is the base pair. So 0 base pair to 2 million base pairs, right? So what will be the conclusion from this figure, right? So we basically cannot draw any conclusion from these figures, because we cannot see the median length of the gene, right? Because the box plot is totally shrinkish by the scale, right? So we cannot distinguish any information about the quantile, median, or anything, the comparable between the chromosomes, right? Why that happened? Because the gene length is skewed towards the 0 base pairs, right? So only few genes have very long base pairs, and most of the genes, they're having like less than 10,000 base pairs. So the gene length has to be around this scale, right? So in that case, you have to choose the log on your y-axis, so you can see the length of your genes in this box plot, right? So you have the log 10 length, right? So 100 base pair, 10,000 base pairs, or 100,000 base pairs. Then you can see that the median gene length is around less than 10,000 base pairs, and the median gene length is quite similar across the chromosomes. So you can easily draw the conclusion by taking log on your scale. So some of the case you have to choose this way, because distribution is always not the normal distribution. Sometimes distribution is skewed in some direction. So if you plot the visualization from your data set, then you have to think about that, whether you go with log or not. So this is another decision point. Or another example you have to think about is the labels. So we can put the X labels and Y labels, and X labels should have clear information about what you put on X, right? And what you put on Y, right? So we're going to continue to look at this one later. Okay. So let's look at some basic usage for ggplot. So you can follow the textbook, or you can go to this link as THDA, which is the easiest things about the visualizations. So in ggplot, you can install the ggplot but you install the tidyverse, so you don't have to install ggplot again, because when you install the tidyverse, it will automatically install the ggplot. So if you want to use the ggplot, you can load the library. So library ggplot, or you can just type the tidyverse, then that will load the ggplot as well, right? So what is the basic structure of the ggplot that we'll cover in here? But before the ggplot in explanation, they explain about the qplot, but don't use qplot, because I mean qplot is quite quickest way you can do plotting, but you know, in the end you're going to use the ggplot anyway, so you know, don't use the qplot. I mean it's way better to exercise with the ggplot. Okay, so ggplot has the basic grammar. So it has the first position for the data frame, or table data frame. Then it has the aesthetic. So AES means the aesthetics, right? Aesthetics mostly has X and Y dimensions. So X means the X-axis and Y means the Y-axis, right? And it has the column name from this data frame. You don't have to put quote like you use in tidyverse. You can just put column names on here, right? So WT is one of the column name from this data frame. So you put WT and you put MPG, then you can get X and Y information here, right? Then once you get that information, then you have to choose the plot type. So this case, we use the geom point, right? So we want to use the point plot. So if you want to use point plot, then you say geom underscore point. So all the ggplot plot type is following these rules. So geom underscore something, right? So let's say geom underscore, then how many plots we can use? So geom bar, this is a bar plot. Geom density is the density plot. Geom histogram plot. Geom point is the point plot. And geom rectangular is you put the square, the tile system plot. And geom violin is the violin plot. And there are also famous one. Geom line is the line plot, right? So those types of plot is the basic things you might use frequently in the exercise. Okay, so once you set up that plot type, then you can specify the parameters to change the plot details. So this plot gives you the basic point plot, but if you change some parameters, like you want to set up the size of the point is 2, then you can increase the point size, then you can change the shape, and you want to use the shape number 23, then it will give you the shape, you know, the blank diamond shape point, right? Not the circle one, right? So you can change that information. Or sometimes you don't specify y-axis, you can just specify x. So here is, you use the weight column from this data set, right? Then you draw the density plot, because density plot will calculate the cumulative functions from your data set, right? So y-axis is automatically calculated by ggplot, not by you, right? So those case, you can only use the x on the aesthetics, right? Or you can use the color information. So let's say this is from the height data set, I guess. the same weight. Then you can plot the density by different sex. To that case, you can set up the field, then you put sex, which is the column. Then you can divide that information too. So you have two ways of using color. One is color and the other one is field. So color, I have to show you in this way. Genome density. Oops. Okay, so if you do that, if you use color, then you can put colors around the shape outline, right? But fill is you get a fill on here, right? You get a fill within the object, right? So that's difference. So this is good, but some density is omitted by other shape, right? In that case, you can adjust alpha. Alpha makes a 0.05. So we're going to put 50% transparency. In that case, you can see others as well, right? So this way you can use information. So what is the difference is the parameters within aesthetics or without aesthetics, right? So how about using here without aesthetics? So ggplot give you error, species not found. The reason is that aesthetics is the one you get information from the database, right? Data frame. If you not use aesthetics, the ggplot cannot find information from the data frame. So if you want to use the data frame, and if you use the column information, that it has to come from the aesthetics, right? So you have to set up the parameters in aesthetics. So you can use category information out of your data frame. Okay, so that was very brief things about ggplot2, and we're going to go through further about the ggplot2 usage using other biological data set in a class time. And this video will cover the following Monday class and probably the Monday after the week. And then we will talking further in our Wednesday class in 5th October. Okay, so hope you have all safe holidays, and also hope you prepare portfolio on time, and see you on the class time. Thank you.